Sure, the curriculum, yeah.
Yes, we need. If you have thoughts on that, it would be nice. Hello, everyone. Good morning, good evening. Can you please type in the chat where you're calling from and what time is in there?
Norway.
We're almost at the weekend here. San Jose, Sydney, 6 a.m. Nice. You see, like it's very cool because I mean it's the whole world. India, it's past midnight there. Thanks for joining, Rag. Cool. Ithaca. Very, very nice. So today we have the big pleasure of being joined by Professor Judith Klinman from UC Berkeley. And I am so in awe of what she has accomplished. She is a pioneer. She's a chemist, and she has almost single-handedly brought the phenomenon of tunneling to the forefront of chemistry, of the forefront of enzymatic functioning. So if you read the chapter of the book, which chapter is it, folks? Alessandro, Natsumi, Abazalt, which chapter is that on Life on the Edge? Chapter 3?
3, yeah.
If you read chapter 3 of Life on the Edge, you will have heard of Judith, and she has really unambiguously shown that both proton and electron tunneling are essential for enzymatic functioning, right? And everything works in nature because of enzymes, or things are made much easier because of the working of enzymes, and enzymes only work because of this funky quantum phenomena. So it's really my pleasure to have the pioneer of this field here with us. Thank you, Judith, for joining us. We have a very interdisciplinary audience from UCLA, from all over the internet, as you saw. And again, thank you very much. And feel free to, the floor is yours.
All right, I have to share screen. Let's see, can I share screen? Oops. Mm-hmm. Okay. Let's get okay. Good morning, good afternoon, good evening, middle of the night, whatever it is. I'm glad to be here. And it took some scheduling, but here we are. Let's see if I can move this over. Maybe move my viewer over. I'm just trying to get it so I can actually see all of the see all of the slide. So give me a second to get this organized. Yeah. What I'm going to tell you today is a little different, perhaps, from what you expected. And now I've lost the pointer altogether. Oh, Lord. Very strange. I'm going to quit and start again because I don't like what's happening. I can't even seem to work the stop share. Okay, how do I get my cursor back? I don't know what I've done. Let's try again. Okay. Yes, all right, let's do this first. Okay. All right, much better. 7, 9. Okay. And pointer. Oh, so bizarre. I really do need the pointer. All right, I'm sorry, I need to stop share again. Try it one more time.
Can I stop share for you? I'll stop share here
don't
by
have a
showing something.
Yeah, I don't have a pointer is the problem.
At some point we could see your pointer.
You could, but I couldn't.
Oh, I see.
I don't know why. All right, can you see my pointer? I'll live with it. Can you see my cursor?
Yeah.
Okay, we'll live with it. Let's go back, reset. And here we go. Basically, I felt that to talk to each of you, I needed to give you some history about enzymes. I didn't think I could just jump into the middle of quantum biology because it wouldn't make much sense. So let's do some history first, and then we'll incorporate the quantum effects. Oh, for God's sake. So now it's not advancing. Oh, I haven't had problems like this in a very long time. Let's see. Yes. Okay. So the first question is, why should we study enzymes? Well, enzymes are really the workhorse of the cell. And, they do every reaction that is important, which includes taking all the food that we eat and converting it to CO2 and consuming oxygen in the process. We're little combustion machines. And enzymes are central to the above, but really all the transformations. And in fact, make life possible as we know it. So they're highly specialized and they're proteins, and they function in water at neutral pH and fairly low temperatures. So the study of enzymes is enormously practical. One, many diseases are due to an insufficiency of a key enzyme. Many drugs are directed toward a particular enzyme. And of course, there's a long-range goal of protein design to produce novel catalysts. But unlike other macromolecules that are central to biology, we don't have a satisfactory theory that relates function to structure. How can that be? The first enzyme was crystallized a very long time ago, and we still don't have that core theory. Here it is, 1926. The first enzyme crystallized was urease. And as of 2020, there were more than 100,000 3D structures for enzymes in the protein data bank. So we have all these structures and we can't reproduce function. Well, the first thing is proteins are large. They may start at around 20 kilodalton and go up to 100 kilodalton in size. That's shown on the left in gray. So basically a protein is a combinatorial series of amino acids which are strung along in a chain and then fold up to give us secondary and tertiary structure. And here what we're seeing is the folded tertiary structure of an enzyme, where the yellow would be one substrate and the red another substrate. So it's a two substrate enzyme that are going to react together. We see that the yellow substrate is bound in the middle of the protein. The red is going to add in subsequently. And the portion of the enzyme that is occupied by yellow and red is very, very small. What in the world is the rest of the protein doing? The active site is tiny in comparison to what nature has provided as an enzyme. Oh, come on. I cannot advance again. I don't have
to unshare the screen.
Oh, you can try it. You can. I think it's when I delete the pictures of the audience and myself. Unshare. We'll share again. I won't mess around. I'll leave everything there, and just have to live with it. Okay, so I can now advance. No. Huh. Oh, for goodness sake. I'm sorry, folks. Something is seriously wrong. I haven't a clue. I usually Zoom at least once or twice a day, often with share screen. So has anyone had this problem before? I don't know. Let's go up. Remote control. No, I don't. All right, now I can do it. Okay, here's our history. We're on the history trail. So Linus Pauling, a very famous scientist from the 20th century, had a hypothesis. And he was interested in antibodies, part of the immune system, and he found that antibodies bound their ligand, their antigen, very tightly. And he said, well, what if enzymes bind part of their reaction coordinate very tightly, i.e. the transition state of the reaction? This is all in the context of transition state theory of reaction progression. And just think of the transition state as an analog of the antigen and have the substrate bind more weakly. So this is expressed in a thermodynamic box, which I'm showing here, where you can think of a reaction as occurring in the absence of the enzyme, the top part of the box. The enzyme is just added in to maintain material balance, and you have the substrate going to an activated complex or a transition state. And then on the bottom of the box, you have the substrate binds to the enzyme first, and it goes to a transition state. The top is the uncatalyzed reaction. The bottom is the catalyzed reaction. You allow the substrate to bind on the left and the transition state to bind on the right. And then you just go around the box in either from left to right along the top or left to right along the bottom. You rearrange the components and you get the expression on the right, which is the ratio of a catalytic process with an enzyme, Kcat, over the rate constant of an uncatalyzed reaction, the same reaction, is equal to the binding of the transition state, this elusive species, divided by the ground state binding. They should be equal. It's a textbook description of enzyme catalysis. So stop, think, what does this tell you about enzyme catalysis? It's just a thermodynamic description of catalysis in general. It's generic for any catalyzed reaction in the context of transition state theory, which is what you learn in freshman chemistry. It does not provide any physical basis for understanding enzyme catalysis. All right, scratch that. even though if you go to your textbooks as you move along in school, you'll find that is the default explanation for enzyme catalysis. Now, very smart man, Bill Jenks, came along and he was very interested in entropy. And his thought was enzymes function by reducing the entropic barrier for catalysis. So what does that mean? Well, here's our little active site indicated by S in the middle of this very large protein. And let's say that whatever is needed for substrate to react, another substrate or a base or an acid or whatever, is close by. And let's say there are many things that need to be close by for the reaction to occur with optimal catalysis. So the reaction can be bimolecular, termolecular, etc. The order of the reaction can be very high. It can involve a lot of components. In solution, all these components would be free in solution. They'd have to find each other. There would be a lot of entropic barrier to getting everything together, so it would work. But on the enzyme, things are prearranged when the protein folds, and therefore you can reduce the entropy a great deal. That's the idea. Now, I don't think each of those components will lead to a large rate acceleration from entropy, from the entropy of activation. But here's a simple example of how putting two substrates together and either having them be free, so they have to diffuse together and then react, and making that the frame of reference, and then comparing it to the same components that are covalently joined, you can get a 10 to the 5th rate acceleration. So a simple comparison among model reactions indicates fairly large rate enhancements from a mobilization of functional groups. And so that could be just the co-binding of two substrates close to each other, confined by the active site. And that's also in the standard textbooks. Now we're not going forward again. I don't know what to say.
Do you want me to stop sharing again?
There is something that's going on, because if I play around enough, but maybe the easiest thing is when this happens, stop share, I'll share, and we'll just go on our merry way. How's that for a strategy? Yeah? If you can stop share, I will share. Yeah, let's see. Still not working. Unbelievable. View window. Let's see what it is.
Do you want me to stop sharing again?
I'll try it myself, perhaps. Yeah, oh, I can't. Then I lose my cursor at the same time. Okay, why don't you do it? Do you want to do the share yourself, Clarice, and see if when you do it, if anything looks, can you share for me? Or do I, you could do it, you can share. You're the host, so you could share.
I would need your slides.
Oh, yeah. All right, forget that. All right, just let me do the share again then. Does anyone have any suggestions?
Folks, anyone? There, we see. We see it change.
I got it to go. It just freezes. Okay. All right. So entropy, that's an important factor. We're getting a little closer to the physical reality of enzyme catalysis. Now we need to step back and ask the question, what is the magnitude of enzyme catalysis? Now that brings us to the work of Dick Wolfenden. And Dick, a wonderful scientist at UNC, figured out a way to examine a whole bunch of different reactions by either putting them in water or reacting them with the enzyme. And at the very top of the figure, you see all the catalyzed reactions, and then the uncatalyzed are on the bottom. And you can see all the catalyzed reactions are about the same. They're all in the range of per second or second order rate constants of molar per second. However, the uncatalyzed reactions have a huge range of difference. And if you just go from the top of the bar to the bottom of the bar for each of these reactions, you get rate accelerations of anywhere from 10 to the 10 to 10 to the 25. And this is an older slide. And actually, we now have an upper limit for enzymatic rate accelerations of 10 to the 30-fold. That's, the number of stars in the universe. So ask yourself a question. Can a static protein structure differentiate between the transition state and the ground state, which are expected to kind of look like each other? No. Can you stop share? We may have to do this between each slide. I think it's related to people joining the waiting room, entering, but okay. So let's go further with a little bit of attempts to use the idea of Jenks, which is that it's all in the entropy and a reduction in the entropy of activation. And, or at least a lot of catalysis happens that way. And that leads us to the work of Peter Schultz, another very well-known scientist, who started to look at antibodies. And he puts together Pauling's idea, enhanced transition state binding, and Jenks' idea of reduction in the entropy of activation. He raises antibodies to small molecules that, as you would to an antigen, antibodies emerge in response to the presence of antigens in the body. And you can do this in ex vivo by exposing antibody production to added antigens, and you get a specific antibody that will bind the added antigen. Okay. And so what he's going to do is add an antigen that looks like the transition state of a reaction that they want the antibody to catalyze. And in this case, they're going to take a carbonate that's shown in the middle. The transition state, the substrate is 1, that's a carbonate, and the transition state for the carbonate hydrolysis is tetrahedral. It starts as sp2. Trigonal goes to tetrahedral at the transition state, and then you cleave and you get a product that you can assay. And he asked the question, okay, how big is kcat? And what's the ratio of kcat to k uncat? And it's not even 10 to the 3-fold. He did the same thing with another reaction, once again using an analog of the transition state to elicit antibody production. He then exposed the antibody to a prodrug, let it go chug along. And he looked at the rates. The KMs are like sub-millimolar. The kcat isn't very fast, not that slow either, but the kcat over k and uncat is about 10 to the 3. Not even close to what we're expecting for enzymes, not even close. So this was supposed to be validation of all you need is tight binding of a transition state. And I suppose proximity of a particular functional group to the substrate. So we could go on and on why it doesn't work, but let's not waste our time. Anyway, we've lost time with all of these technical problems. So now we get to de novo design. And David Baker, very successful computational chemist, has gone through the development of protocols. Once again, you can go to the literature and read for yourself. But, and he uses as a scaffold, in this particular study, it's called a Tim barrel protein. 10% of solved enzyme structures are for these Tim barrel proteins. They're all over the place. They do all kinds of reactions. And he decides, okay, what I'm going to do is look at this reaction. I'm going to try to cleave a carbon-carbon bond that doesn't occur normally in nature, at least as shown. And that what we want to do is have the classical Tim barrel enzyme do this reaction. We're going to figure out what we need in terms of functional groups, and that's in the prior set of commands that he used in the computer. They worked very hard at this. And they come up with different motifs for the active site and mechanism. And here are their K-cat over K-uncat ratios. A little better than what we saw with catalytic antibodies. 10 to the 2, 10 to the 3, 10 to the 4. Okay, one more step, and that's called directed evolution. Here's Francis Arnold, Nobel Prize a couple years ago with Don Hilbert. And what they're going to do is take that same, it's called a retroaldehyde, and they're going to subject it to directed evolution and see what happens. And they do it many rounds. And it gets better. So directed evolution, and we don't understand how directed evolution works. Francis thinks it's too hard to figure out. I think maybe the information is there, but here's the result. You actually get as high as 10 to the 7, 10 to the 6, 10 to the 7-fold. For this design enzyme, and then the subsequent application of directed evolution. So directed evolution is doing something. And this is important for us because the mutations that arise on directed evolution are not in that very narrow strip. That's the active site, the active region of the enzyme, but they're all the way out, distributed throughout the enzyme. So here's a summary of where all this has led us. So if you look at the tan, you get the range of rate accelerations, this K-cat over K-uncat for the computer design, maybe as high as 10 to the 7. If you then apply directed evolution post computer design, you're optimizing with directed evolution, you may get up to 10 to the 10. But red is where all the native proteins in nature exist. What is going on? We're looking for new properties and concepts. So let's go then for the rest of the talk, second half of the talk, to quantum biology. We're going to tease apart the missing factors that have limited our understanding of enzyme function. These new concepts can provide new guidelines to the successful de novo design of highly active catalysts, either enzymes or other soft materials. Enzymes move, they're not static, they're moving all the time. They either undergo large conformational changes on the bottom right, they undergo domain shifts, undergo loop closures, they undergo subtle rearrangements. These vary anywhere from seconds to picoseconds, really. And the distances of the movement can be anywhere from 4 angstroms, as illustrated, to 0.02 angstroms. We're particularly interested in the nanosecond, microsecond, nanosecond, picosecond motions. The others can be observed. We can see them in X-ray crystallography and by other methodologies. But it's in the elusive range that we want to know what's going on. So the first thing is, one of the missing features is almost certainly in the area of dynamics. So structure goes to dynamics. What is that dynamics and how do we relate it to function? And the other thing is, we have always treated the reactions classically, transition state theory, and what's been missing is quantum mechanics. And we have been focused primarily on hydrogen tunneling. And this is a representation of calculated wavelengths using the de Broglie wavelength for small particles. You can, which then assumes the interconversion of particles and wavelength and waves. You can calculate the wavelength, and the wavelength is very mass dependent. And in fact, I'm showing you the example in the blue boxes where the total energy of the system is 4 kcals per mole. So, the calculation of wavelength depends on the total energy of the system. As you can see, it will change. If you just go one tier down, you'll see how the wavelengths get smaller as the total energy goes up. Nonetheless, this table shows some very important things. First of all, the electron. The electron has a very large wavelength, close to 30 angstroms. And electron tunneling is all over the place in biology. I think most people accept that, if not everyone. It's the nuclear tunneling, which people have been more, they have had a lot of difficulty accepting that making and breaking bonds, not just transferring of electrons, can occur quantum mechanically. And you can see that the wavelength for the proton or hydrogen is much, much shorter than the electron, but the difference in size is about 2,000-fold. And then you can see that if you put in the isotopes of hydrogen, deuterium and tritium, the wavelength just gets shorter and shorter. But the key feature, and this is what is shown in the bottom in red, the key feature is the wavelength of the particle in relation to the process that's taking place. So for electrons, very often the donor and acceptor are 20 angstroms apart, and the wavelength is long enough to achieve the tunneling, and there could be hopping, but nonetheless, that's suited to the distance of 20 angstroms is suited to long-range electron transfer. When you get to hydrogen transfer, if you look at all the modeling of proton hydride hydrogen atom transfer, generally the distance that the particle is moving is 0.7 angstroms. And so it's the relationship of the wavelength to the reaction that's taking place. When they are in the same regime, you cannot ignore wave function overlap. All of a sudden, we have a new door to explore catalysis. Furthermore, about 1/3 of enzymes catalyze CH activation. And decades of work now indicate conversion of reaction to product involves movement of hydrogen as a wave. Now, if that's the case, you'll see how this becomes really important. The barrier to catalysis comes from the heavy atoms of the protein. This large gray scaffold starts to light up. Something's going on there. And in contrast to electron tunneling, we have isotopes of hydrogen as a powerful probe. And because deuterium is heavier than protium, its wave function overlap, as shown in this picture, is less than for protium. And this focuses attention on barrier width. Remember, transition state theory, it's all barrier height. Now, if we look at properties of hydrogen transfer, there's something called semi-classical theory that has a quantum component, and that's part of transition state theory. So shown above on this slide is a typical reaction coordinate where you go from ground state to transition state. But the ground state, this would be a CH cleavage reaction, is comprised of a vibrational level. We can think of it in terms of a single vibrational level. To simplify, we have a CH bond or a CD bond or a CT bond. And so these are quantized. The vibrational levels are different for the different isotopes, and it comes from the difference in reduced mass in the stretching frequencies. So there are isotope effects without quantum, what we call true quantum effects, but still, this is not purely classical, it's called semi-classical. If we then look at the rates of the reaction, what you see is because this difference among isotopes disappears at high temperature, the difference in vibrational levels gets smaller and smaller, and you plot ln K versus 1 over temperature, you extrapolate basically to the y-axis. And so the isotope effect essentially goes away in infinite temperature. And there are lots of predictions, which is that the isotope effect based on this semi-classical model, give or take a little bit, it's 7. And then you can look at the relationship of H to T and D to T and make predictions, and that's shown. There's an exponential relationship, which we're not going to focus on today, but for reference, it's 4.5. And then you can look at the intercepts, the Arrhenius prefactors, and it's between 0.7 to 1.2. It's about 1. And the Arrhenius prefactor itself is 10 to the 13 per second. That's the rate of a bond vibration. And what do we see in enzymes? Here we go. We see isotope effects of 700 at room temperature. The exponential ratios, as shown, they go up to 16. The isotope effect on the Arrhenius prefactor goes up to 700. The Arrhenius prefactor itself goes up to 10 to the 23 per second. And here's a summary of some of the isotope effects and temperature dependencies. I don't want to, but and there are many, many more than this. This is just an example. Okay, so here we are. Lots of new data, totally different behavior from what we expect for semi-classical behavior. So there on the top A, I've just reproduced the semi-classical behavior with 0-point energy contributing the isotopic differences in reactivity. On the bottom, labeled B, we've got full tunneling, where the particles move completely through the barrier. And in this case, there is a temperature dependence for the reaction, but there's no isotope effect on the temperature dependence for the reaction. That is, the difference in activation energy for D and H disappears. Very curious. Well, B is what we see in most native enzymes, and it's not precise. Sometimes it's just small temperature dependence. Many times delta E, delta EA is in fact smack on 0. And so here are the kinds of things we see. There's a review in Accounts of Chemical Research 2018 with Adam Offenbacher. So on the left, native behavior, no isotopic effect on the energy of activation. What's that mean? And then if we get moderate impairment, like putting in the wrong amino acid, an altered amino acid at the active site, all of a sudden it starts to look semi-classical again. And then if we put in an even worse amino acid, the lines start to cross and we get extreme impairment. And now EA for deuterium is much larger than EA for protium. And we see this trend in behavior a lot for many different enzymes. Let's look at a real system at the time. And this is lipoxygenase. This is a very studied enzyme. It's a paradigm for extreme tunneling in enzymology. And some of you may have heard this before, but let's go through some of the key experiments. This is a large enzyme, about 90 kilodalton. And it's got one cofactor. It's an iron in the +3 oxidation state. It's bound to a hydroxide. That's the active form. It's labeled red and white in the middle. The substrate, shown in green, has been modeled in. That's not from a crystal structure. It's been minimized. We'll talk about that later. The black is the reactive carbon at C11. And then there's some hydrophobic residues that are very important for what I'm talking about. There's Lu 754, Lu 546. These are hydrophobic residues that reside on either side of the reactive carbon-hydrogen bond. We're going to break a carbon-hydrogen bond. And then one helix turn away, there's Lu 553. Now, this is a hydrophobic substrate, except it's linoleic acid. It's a fatty acid. It's got one charge in it at the end, a carboxylate. Okay. And the data for this slide represent mutagenesis studies at position 553. And there's the energy of activation, the isotope effect, in this case 81, going up to almost 200. Now note, in terms of mutants, this is very important. We don't put in charges, or we don't change charge or hydrogen bonding or anything like that. All we want to do is change the interior packing of the substrate. It's the most modest perturbation we can make, and see how that shows up. We're trying to understand the role of the scaffold. And so you can see the isotope effect stays the same pretty much until we get to glycine, which of course has no side chain. But then if we look at the difference in activation energy, the temperature dependence of the KIE, it's not 0 for wild type, but it's small. And then it gets progressively larger the smaller the side chain at 553. We're creating more and more space in that region where you really have an ILU. And you can see the trend. Okay. And we have low temperature X-ray structures. The mutant enzymes are unchanged. So what is going on here? And this took many years. I mean, Kuznetsov and Olstra published a paper in 1999. We incorporated that in 2002, and then Sharon Hammer-Schiffer came along in 2010 and gave a really solid analysis that it comes up with the same result we had, but I think is really very elegant, and et cetera, et cetera. You can see these references. If you want to refer to a particular reference, I suggest the third one, Hammer-Schiffer and Sudakov in Chem Reviews 2010. Basically, we have analytical expressions that will accommodate the behavior. Now, what we're able to accommodate is the temperature dependence of the KIEs, which is what is so bizarre. And we start with electron transfer theory, Marcus theory, which is well developed, and it really depends on the, I can't use my cursor, but if you look at 2.2 on the right, there's an exponential term that has a temperature in it. It's the major temperature dependence of the rate. And it's got the environmental reorganization energy, lambda, and then the delta G naught, the reaction driving force. And then it has temperature in it as well. And so those are the dominant features that control electron transfer together with the electron coupling. However, when we're transferring a proton and an electron, and you can think of, so basically in the oxygenase reaction, there's a hydrogen atom transfer where the proton goes onto the hydroxide of the ferric hydroxide and the electron goes onto the iron. So you can actually separate it out and call it a proton coupled electron transfer. Now what's going on here is both the electron and proton are tunneling. And this becomes non-Born Oppenheimer behavior. And instead of just looking at electron coupling like you do in electron tunneling, you've got electron coupling, of course, you're transferring an electron, but also the wave function overlap between the hydrogen in the donor well and the hydrogen in the acceptor well. So that's called vibronic coupling. It's actually non-adiabatic because it's non-Born Oppenheimer. And it's electronically non-adiabatic. I know this sounds like a lot of different terms, but essentially rather than the electrons moving faster than the nuclei. In this case, when the hydrogen is tunneling, the hydrogen is moving faster than the electrons. They're all coupled together, and that's the V sub UV term. U being reactant and V being product. Absolutely essential to understand what's going on in these H tunneling reactions. Now, in the top, 2.2, the donor acceptor are considered fixed. Protein may be moving, but that equation is a static one with regard to the protein and the substrate in the protein. However, we know that, as I showed you before, protein is moving all the time, and the donor and acceptor are expected to go and sample all kinds of distances. And that's 2.3. That's the vibronic overlap as the hydrogen donor and hydrogen acceptor start to sample distances. So now we have a direct probe for motions within the protein. And this is able to fit beautifully the available data for the oxygenase. One thing that's worth pointing out is that if you just calculate the rate and not the isotope effects and their temperature dependencies, basically what you see is that rates are much too fast. The K tunnel you calculate is much too fast. And that's because at any given time, the fraction of enzyme that reacts is very small. E total is not what reacts. Some very small fraction of E total is what crosses the barrier, which is important. So just to take a look at this visually, this is really a central part of what's going on. And here's a picture where we start with a bound substrate. A lot of it will be in a, if you look at A, a lot of the ES complex may not be active. It has to rearrange itself to go into the blue regime. That's A, the active positioning of the substrate. And then you have dynamic sampling and you get a little more activated. And then that's part of the conformational landscape. And then that green region has to undergo the environmental reorganization, lambda, in the equation, Marcus expression. And that lambda is what brings transiently the energy of the reactant and the energy of the product into degeneracy, transient degeneracy. That's shown in C. There's your wave function overlap. And then it breaks. It can break where either the hydrogen's on the left or on the right. Now, what you see is A and B is just movement of the scaffold. It's going to be temperature dependent, but independent of the isotope in the substrate. C, which is the wave function overlap, is temperature independent. It's quantum mechanics. It's quantum mechanics. It's not temperature dependent, but it is very dependent on the isotope. So you've got two coordinates now. You've got the isotope dependent coordinate, which is C, and the temperature dependent coordinate. And this leads, if you go over to D, you have two pictures there. One is native enzyme on the bottom and one is mutants. And you can see for the native enzyme, you've got two coordinates that are acting orthogonal to one another. And that's why you get parallel lines. So you get the same activation energy, right? That's independent of the isotope at the reactive bond, but different isotope, but different rates, because the amount of wave function overlap is much greater for protium than deuterium. Bingo. Now, we can also explain the trend through mutation, but I don't want to spend time this morning because there really isn't that much time. And the other feature of all this is we can get the donor acceptor distance, the most probable donor acceptor distance for effective wave function overlap. And that's 2.7 to 2.8 angstroms. And there's a lot of question of, how does the enzyme generate this short distance? I don't think I have time to go into this. We have measured the distance using C13 labeled substrate and the paramagnetism of the active site metal. don't think we need to go through the details. And the equilibrium distance is as expected for the donor and acceptor. It's 3.1 angstroms, van der Waals. And we have to get to that tunneling distance, which is shorter by 0.4 angstroms. And the question is how. So we need to think about now the fact that SLO, lipoxygenase, is thermally activated, as are 99% of all enzymes. So we have to think about thermal activation. And how do we get then this thermal activation to correlate with this reduction in distance? And so we have a lot of tools that are possible, but I want to introduce a new tool that we've developed over the last 5 years, and it's called hydrogen deuterium exchange. So it's hydrogen deuterium exchange by mass spectrometry. Mind you, our goal here is to understand the role of the protein scaffold and the dynamics within the protein scaffold that are thermally activated and can be related to the catalytic event. So we take our protein and we incubate it in D2O. And what this does is lead to an exchange of the amide bonds. Within the chain, there are a series of amide bonds linking one amino acid 1 to amino acid 2 to amino acid 3. And the protons on the amide are susceptible to exchange by deuterium when you incubate in D2O. So you let it sit in D2O for a given length of time. And then you quench. And you quench by going to low temperature, 0 degrees, low pH 2.5. And then you use a protease that works at low temperature and low pH. And you chop the protein up into these peptides, which are usually about 12 amino acids long. And then you look at the incorporation of deuterium that's occurred as a function of time. The longer you incubate, the more deuterium, usually into the peptide, and temperature. And that's new. People have not focused on the temperature dependence, but we're trying to get at thermal activation. And so if you look at the rate expression, the rate at which this happens at any single temperature is given on the top. K open, K op, is just the rate at which the protein undergoes some local unfolding, which allows the D2O to come in and exchange with the amides, multiplied by the actual chemical exchange of protein by deuterium. And in the numerator, we have K closed, which is the rate at which this partially open protein comes back together, plus K exchange. And if you can find conditions, which we call EX2, where the closing of the protein, it's open, that's the unnatural position of the protein, closes much faster than K exchange. And so then we get the rate of deuterium going in to replace protium is equal to an equilibrium constant. It's this opening and closing times the rate constant for the chemical exchange. And that means that the enthalpy of activation for this exchange, because now we're going to look at temperature and get out EA, which is related to delta H observed by just RT, is comprised of a thermodynamic term, delta H naught for K op over K closed, and a kinetic term, delta H double dagger. Now, delta H double dagger is well established with small peptides. It varies a little bit depending on the structure of the peptide, but it's in the range of 17 kcals per mole. Okay, so for lipoxygenase, SLO, 94 kilodalton protein, do what I just said, we get a huge number of peptides. This is Adam Offenbacher's work. Get 49 non-overlapping peptides and 89% sequence coverage. And then you measure HDX as a function of time, temperature, and functionally impactful site-specific mutants. Well, I've already talked about 553 as being functionally impactful. We know 746 and 754 and 546, which sit on either side of the reactive CH, are very functionally impactful. And then we look at the difference in the enthalpy of activation for the wild type versus these mutants, right? And we already know what the impact of these mutants is for the energy of activation for catalysis. We're using delta H and energy of activation interchangeably. It's fine because we're always looking at differences. And so when we do that, after we've mutated, that delta H double dagger term down on the bottom in black drops out because that's not going to be affected by mutation, because the it's at one position, and we're looking at all these peptides, none of which except for one peptide has the site of mutation, and it's unlikely that that's going to have a very big effect on the intrinsic rate of exchange. So what we get out is the effect of the mutation on this local opening and closing. So we get the effect of mutation on the temperature dependence of this local opening unfolding. And to go one step further, then how a mutation that alters the activation of tunneling in a distal region of the protein correlates with the impact of that mutation on the opening and closing of a protein at the surface. Here are the results. So on the right, on the left top, you see the box in orange, and that just shows you the region where we see an impact of mutation on the opening and closing through HDX, hydrogen deuterium exchange. And the dominant peptide, it's just two peptides, and the dominant peptide is 317 to 334. So if you look at the right on the top, you can see the orange ball is the iron. The two residues that are around the reactive carbon are 754. That's in blue for a good reason. I'll tell you in a minute. And in red is 546. And another red residue is 553. So when we first did these experiments, Adam saw a one-to-one correlation between the energy of activation for catalysis, which we knew before, and the energy of activation for HDX. And if we look at the graph, the red line, you can see wild type in the middle, and then the energy of activation for 546 went up, both for HDX and KCAT, went down for I553G, and we have a new mutation that we added on, 552A. So we have these four examples, 3 mutations plus wild type, that fit on a line where, as the energy of activation goes up for catalysis, so does the energy of activation for HDX. 754, which is on the other side of the reactive bond, has a peculiar and important behavior, which is that it affects KCAT, and we understand why now. It has to do with the properties of the active site, but it does not affect the energy of activation for HDX. And if we make mutants, double mutants with 754A, whatever we see depends on what the double mutant is comprised of. But if we introduce a side chain which is shown to alter KHDX, we get an effect that reflects that without the 754A, i.e. 754A lies off what we call a thermal network. We define the thermal network by the line red and the red residues, and the blue, because of the lack of sensitivity of the energy of activation for HDX to 754A, we call that off thermal network. So we have an internal control for what's going on, which is pretty close to the R, but not on the thermal network. Okay, one thing you want to notice, these are not identical values, they're just correlated values. very different chemical processes. KCAT is wave function, the activation of wave function overlap, and HDX is activation of this opening and closing of the protein in a particular region near the surface. And this is the thermal network. You can see how we have 546 and 553 are definitely on the thermal network. 552 is on the thermal network. And then you can see the presence of a helix, loop helix near the solvent. And we could talk about this a lot, but I wanted to just move on and say we've done further studies using a technique called room temperature X-ray crystallography. And the final thermal network is shown on the right, where you can see we start with the bound substrate and modeled once again, and we have carbon-11 within van der Waals of 546, which seems to be the key residue, which then radiates out to 552. That's also been shown by HDX to be on the network. And then you have all these van der Waals interactions. It's like a cone that reaches from this point out to the protein-water interface. So that's where we are with spatial resolution. Time is gone. So I want to just say that I could tell you much, much more, and I won't because of a lack of time. But I want to just say that this behavior is seen in many proteins. And we have, instead of just HDX experiments, we have very extensive real-time kinetic experiments where we can measure time of protein motions through fluorescence studies, Stokes shift studies, FRET, etc. So we've complemented the HDX studies, which are to locate these thermal networks, with kinetic studies to look at the time constants for the activation of the thermal networks. So the first thing to close up I want to show you is a picture of the mechanism of lipoxygenase that we are left with, the tunneling mechanism. And this is a result of not just the HDX studies, but also the very detailed kinetic studies, where we have complementary results indicating greater than nanosecond activation of this region of the protein, a cooperative activation of protein in this region. So here we are with the iron hydroxide again. And you can see carbon-11, you can see 546. Here's our network that I already showed you. And the idea is as follows. Basically, heat is going to flow through a protein in a very anisotropic way. And if we think just about lipoxygenase, what it's going to do is travel through, reaching the keystone side chain. If you look at the top, carboxylate in, use that as both models, carboxylate in and out work, but carboxylate in is easy enough to see. And the heat reaches through to 546, which is in van der Waals with carbon-11. And what this does is result in a shift of carbon-11 toward the hydroxide of the iron. There it is. That's how we get from van der Waals, 3.5, from modeling 3.1, from our Endor experiments, to the 2.7 we need for tunneling. At the same time, this pulse of heat will move the substrate in its entirety into a different position. And you can see carbon-14 for this orientation is in van der Waals contact with I839, which is the C-terminal residue of the enzyme and a ligand to the iron. And by so doing, forcing a hydrophobic residue toward a charged carboxylate, change the electrostatics of the hydrogen acceptor, thereby facilitating the the energetic degeneracy between reactant and product wells, which is a prerequisite for tunneling. Boom, you've got this transient push, and you've got everything you need for tunneling. And of course, the number of times it's successful is relatively small. Now, there are many, 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 many things I can talk about. And I didn't know how fast I would go. But basically, I'm going to just sum up with a picture, if you will, which is a composite, not just of the work on SLO, but overall, where, as I said earlier, the observed rate for tunneling is much, much slower than the calculated tunneling rates. And that's because the probability of reacting is very small. So so the extended model can go as follows. The thermal energy from bulk solvent is deposited into specialized networks. And the rate constant has to be faster than the fastest measured process. I didn't have a chance to show you those data, which is greater than a nanosecond. Experimental and theoretical studies support heat transfer through soft materials in the range of 10 to 20 angstroms per picosecond. Now, the distance between the solvent and the ferric hydroxide is about 20 to 20 angstroms. So site-selective heat flow in these thermally activated enzyme reactions will produce a local out of equilibrium behavior relative to the total system, which of course has to be fully equilibrated at each temperature, which means some regions of the protein are hot and some are cold. So I won't spend a lot of time on that, but the implication is a coevolution of embedded site-selective thermal conduits along with the active site structures, which is where we started, cofactors, as the origin of the remarkable catalytic efficacy of protein catalysts. In the left, we see just isotropic thermal energy activation, which would be both constructive and destructive, but enzymes have somehow co-evolved these thermal conduits. And that is an important component of their enormous activity. And we would never have gotten to this picture in a convincing and quantitative way if it weren't for our findings of tunneling and atom transfer in enzymes. And I'll skip all this and just point out the importance of, in terms of SLO, the work of the recent work of Adam Offenbacher and Paolo Zaragoza, and I thank you. And Clarice, we can stop share.
Sure. Thank you, Judith. There is one question. I have a couple of, there is already one question in the chat. Rag asks, is there such a thing as the secondary structure of a protein determining whether we would see more of electron?
Well, the electron transfer, of course, is long range. And Harry Gray has studied this in great detail and found that there's certain protein side chains which promote electron transfer and are an essential, that there are discrete pathways for electron transfer. Let me put it that way. They're aromatic residues for the most part. And so that's quite important. As I said, the distance over which the hydrogen tunnels is very small, 0.7 angstroms. But the key is that it shows all these anomalies. Remember, the distance that it moves generally in all of the theoretical treatments is 0.7 angstroms. The wavelength is 0.7 angstroms. It's moving as a wave. But what that does is the wave property just kind of sheds light on aspects of the reaction dynamics and the protein dynamics that we could not have seen before. So I don't think the secondary structure itself for hydrogen tunneling is critical. I think the overall, or maybe you're talking about thermal networks and whether the secondary structure is important for those thermal networks. That we don't know about thermal networks in electron tunneling. People, if you look at the older literature, Jortner, very famous theoretician, suggested that, but no one has ever had an experimental tool to pursue it. Because in electron tunneling, even though you don't have that donor acceptor distance sampling, because it's much less sensitive to small changes in donor acceptor distance, you still have to get degeneracy, in order to get electron, you have to get the environmental reorganization just right in order to get the right ground states for tunneling. I think we have a lot of ideas about, we have looked now at 5 or 6 enzymes. We have thermal networks for 5 or 6 enzymes. I could show you real fast because each enzyme has a different thermal network. Let me just show you real fast. Let's share again. I have 2 slides. I mean, I wanted to go slowly because I wanted to be able here. So here are the 2 tunneling enzymes. I only spoke about one. Here's the oxygenase, and what you see in magenta is the thermal network we found. And the evidence here is really strong. On the right, we have a hydride transfer reaction, a totally different enzyme. It's a dimer of dimers. It uses an organic cofactor. I mean, it's so totally different. And you can see that there's a different kind of thermal network, which involves residues that, this is a 2-step substrate enzyme, and the thermal networks lead behind each of the substrates. It just kind of merges when you look at it in 3 dimensions. And so you can, the secondary structure, and one is a hydrophobic island in soybean lipoxygenase, and it's more complicated in the thermophilic alcohol dehydrogenase. Now, that's all from tunneling. What happens if we look at enzymes in general that don't tunnel, don't support hydrogen tunneling necessarily? Here is the, we went back to the Tim Barrel, which was David Baker's construct for de novo design. And we took three members of the Tim Barrel enzyme that have the same scaffold, but catalyze different reactions. And here's one in a adenosine deaminase. You can see on the left, looking at the magentas in the catalytic bowl and rotate 180 degrees. That's the back side. The thermal network's right in that catalytic bowl. Then we have yeast enolase, lyase. It's a dimer, so the catalytic bowl is on the left and the backside is on the right. Here you have a loop that closes over the active site, and then you have 2, like a U shape, 2 regions that constitute the thermal network, leading into where the substrate and metals bind. Totally different. And then we have a methyltransferase on the bottom. It's a partial Tim Barrel. And the thermal network is all around where the extended, very large cofactor binds. So I guess we can stop sharing it, but I think that you can see each one has a different property. So yes, I don't know if that's the question you were asking, but secondary structure is going to make a difference in terms of how these thermal networks get constructed, but not in terms of the tunneling. I don't know. I would say not in terms of how the tunneling goes on, but it's certainly in terms of how the heat gets transferred to create the 2 key properties of tunneling, which is to get things closer together, right? And to get the degenerate energy levels.
So Judith, based on that, before I go to the other question, I have a question. When you say thermal network, you mean phonons that are due to a finite?
They're vibrational modes. They're vibrational modes of the protein. Yeah.
So now I have a question. Maybe I want to know if that's similar or different. So I am curious about the phonons brought by bond vibrations, not temperature. Well, they're all temperature dependent, but like specific to protein vibrations. And the analogy or not that I would like to propose is, we know that there is this idea that, say, for maybe olfaction is based on a model that is like an antibody, like it's a lock and key plus vibration, right? That molecular vibrations play a role in the tunneling of the electrons that make the binding of the thing that to give you like olfaction and stuff. I'm not sure I'm
That's
explaining this
interesting,
correct.
because in fact, electron tunneling is going to depend on this anisotropic use heat transfer as well. If it's not, but if it's photochemically activated, that's different. See, we're focused on enzymes, 99.9% of which are all thermally activated. When you get into photochemically activated reactions, it's different.
No, I don't think it needs to be photochemically activated.
But
It's just
for
like
scent, it's different. It's not photochemically activated. For vision, yes.
no. I just wonder if molecular. So, folks, help me here. I'm not sure. Alessandro, you saw that in detail. Rephrase my question. I know that you know what I mean. That I cannot.
I mean, I didn't really get the question either.
You didn't.
So I
Okay.
know that the tunneling process in the olfaction is supposed to be inelastic tunneling, electron tunneling, which is mediated basically by the vibrational modes of the molecules. That's how the electron loses energy and goes to the acceptor spot, let's say, from donor to acceptor. But I didn't get your question.
Okay, my question is, might there, when Judith says enzymes are thermally activated, I think it's like thermal modes having to do with there being a finite temperature and less to do with like vibrational modes of the molecules of bonds. My question is, Judith, do you think that vibrational modes of the molecule might also be playing a role, or is it exactly what you mean by thermal networks?
Yeah. So I don't think that the molecules themselves are playing a role because they're too high frequency. I think that CH stretch is 3,000 wave numbers. I think these are like anywhere from 20 to 100 wave numbers. I think these are low frequency modes. I think they're cooperative, and I think they're intimately related to the what you called secondary structure. That, I mean, I haven't thought about it in that way, but we don't know yet. For example, in lipoxygenase, there's a loop that is at the end of this hydrophobic island. It's called an ILV island. And we have some collaborations going on where we're looking at the impact of mutants on free water and bound water. And our model that's emerging is that it's the free water that's diffusing and hitting the loop that is then leading to this global restructuring, which is the activation energy in that region of the protein. And then it dissipates. And then it happens again, and then it dissipates. But it's not going to be at 3,000 wave numbers at room temperature.
Wait, why not? Because that's exactly, again, Alessandro might correct me, but I think that's exactly the frequency range that might be at play for this model of olfaction. Is this correct, folks? It's
because the model of olfaction has to do with the odorant and the fact that when you deuterate it, you get a different response. I think those experiments are very controversial. I think the model, however, which is the use of the ambient temperature to facilitate electron tunneling, and that can only happen when the odorant is bound. That is, this is all part of allostery. It's almost an allosteric issue. So, for example, we know we get a protein in one conformation, and then you bind an allosteric effector, and it goes like this. And now the thermal networks don't change, but the access of the that's my view. No one's proven that yet. The access of the thermal network to whatever the active site is has changed. I have a series of slides. There just wasn't time to talk about it. So I think that, you know, in a faction, it could be that there are isotope effects on binding. These are known. You know, you can look at a deuterated compound. If you do a column chromatography, it's retarded more than H. It binds differently. So there could be subtle effects that lead to, you know, a little bit of difference, which then alters the ability of the heat to flow, which facilitates the electron transfer. I never thought of it before, but, and as I said, when we started writing all this up, I went back and looked at the early literature, and Joshua Jortner, who is really one of the giants of early electron transfer theory, was talking about this stuff, temperature dependence. I mean, I didn't even catch it until 6 months ago. And, you know, in electron transfer, not in hydrogen transfer. And it could be that the sort of things we're seeing for hydrogen transfer could be conceptualized the same way for electron transfer. It's just you don't have that donor acceptor distance. I don't know. And that could be dependent on secondary structure. But the thing about a CH activation, and I don't know where the deuterium was put in the odorant, but it would be 2,000, 3,000 wave numbers. I'm thinking the frequency for the stretching frequency.
So let me try to understand. You're saying that isotopic substitution could have important effects on the conformation of the protein.
Not large effects, but important effects. I think it would be a subtle effect. Okay. Because we know that isotopically labeled compounds bind differently to hydrophobic resins. Something's going on. So I don't know how that would manifest itself in the protein, but it's an interesting idea. But then this idea of secondary structure. I mean, we have a second protein we're looking at in great detail, and it looks like the secondary structure that affects, it's not a tunneling enzyme. So the whole idea is everything we're seeing with tunneling, getting close, right? Getting short distances, probably the same. Getting the electrostatics just right, probably the same. We're seeing a helix that's amphipathic, hydrophilic, hydrophobic, and here's the substrate. It's a hydrophobic wall, and we think it's the solvent hitting this, coming up against the bound substrate and moving it in relation to the metal hydroxide over here. There's so much here that, you know, I don't have enough time, enough years left to do it, but I think it's not always quantum biology, but what quantum biology did was force us and give us the tools to reframe the important questions. And I think that has to be included in terms of the importance of quantum biology. It has that property, and it has the opportunity to give you precise mathematics. And once you have that, then you can, it's not just hand waving anymore. The first part of my talk was about all the hand waving that went on for 50 years, right?
Judith, there is one final question. Is there any possibility for enzyme stacking in order to increase the tunneling length, or increase or decrease? I'm not sure what it's meant.
Well, electron transfer does that, of course, where you go from here to here to here to here. I don't know about hydrogen transfer, how you would do that. You'd have a very tight stacking because the transfer distance is only, you know, a little under an angstrom. And so you'd have to have, there are many reactions that do multiple H transfer, but that's for electrostatic reasons. You have, you know, two groups like this, and they're connected by proton donor acceptor, and proton goes here, and then the proton goes there, or something like that. But you mean to facilitate something that you want to see? See, one of the applications, I think, of what we're seeing is using these thermal conduits to activate active site chemistry in ways that haven't been thought of before. And some of that could even be quantum events, could be hydrogen transfer to create a new molecule that could be used for signaling or something. I think, but understanding how to initiate through these thermal conduits.
Daniel, do you want that? Daniel asked the question. Do you want to unmute yourself?
I think that answered definitely most of it. Is that with the electron stacking would be easier to make those lengths with the hydrogen where you have such small tunneling. I think that most of it. Thank you.
You know, the non-Born Oppenheimer behavior in this system is in H tunneling is very interesting, because normally you think the electron moves and very fast, and then all the nuclear coordinates accommodate. But in vibronically non-adiabatic H tunneling, the electron's moving more slowly than the hydrogen because it's just wave function overlap. Boom! It's not instantaneous, but it's really fast. I mean, I just think that was a box that chemists had a hard, see, physicists are different from chemists, but that's a box that chemists have a hard time climbing out of. The Born-Oppenheimer approximation, for example, is like, yeah, sure.
Maybe we should end this session with plenty of questions and stuff, but we're over the allotted time of the students. So, Judith, thank you very, very much for this super exciting class. There's plenty of people saying thank you in the chat. And I think you are officially our final lecturer of this class. Next week, there is the final class in which I'm going to do a recap of the course and a conclusion. Judith, thank you so much again for your for your generosity with your time, and I look forward to interacting. We all look forward to interacting with you more in the Quantum Bio GRC.
Definitely, and good luck going forward. Thank you. I'm glad we finally pulled this off.
Yes, yes.
Right.
Thank
Okay.
you so much, folks.
And I don't know what
Bye-bye.
was going on electronically. Bye.
It was tunneling too much. Bye-bye. Take care,
Bye-bye.
everyone. Bye-bye.
Happy holidays.
Happy holiday.